Hey everybody, it's your human anatomy and physiology teacher, Mr. Poser. Today we're going to be getting into the real physiology of the nervous system here. So what we're going to be discussing is the action potential. And an action potential is really it's the process by which nerves communicate to each other. And nerves have to communicate, and neurons have to communicate to each other in order for anything to happen within the nervous system, whether it be brain function or integration, sensory input, motor output, any of that kind of stuff. So yeah, the action potential are electrical signals that are sent through the axons of neurons. And what we're going to be looking at today is down to the nitty gritty, the molecular process by which the action potential happens. All right. So as I was kind of saying before, an action potential is a change in electrical potential associated with the passage of an impulse along the membrane of a nerve cell. Okay, so if we're talking about electrical potential, there's a word for electrical potential. It's called voltage. Okay? It just means there's a difference in charge on one side of the cell versus the other side of the cell. Okay, so what a neuron is able to do is just very briefly change the electrical potential on either sides of a membrane. So we talked a little bit about how membranes work at the very beginning of the year, that they're semi-permeable, or they're selectively permeable, excuse me. So they let certain ions through or certain molecules in and out, and they pick and choose what comes in and out. And that's understanding the cell membrane is going to be really important for understanding the action potential, because an action potential occurs along the membrane of a nerve cell, more specifically the axon, which is what we looked at in the last video. All right, so just as a recap of some electrical terms here, voltage is a measure of electrical potential energy. All right, and the units that we often use for, say, like batteries are volts for voltage. When we're discussing an action potential, when we're discussing an action potential, we're talking about millivolts. So 1 volt times 10 to the negative 6. No, 3. Yeah, 10 to the negative 3, excuse me. That's a millivolt. So we're dealing with less voltage, but just a slight change in voltage on either side of a cell membrane. That's what an action potential is. So what are we even calling it voltage and potential energy for? Well, now we got to get into a little bit of physical science. Battery has electrical potential energy until you connect a wire to the end of each battery, to each end of the battery, creating a current. Okay, so you're converting that potential energy into electrical current when you hook the battery up to the wire, right? You got a positive end and a negative end, and that's an electrical charge difference. Okay, so you hook up a wire to this, it's going to produce a current. Okay, so thus this battery has stored energy, and that's what potential energy is. It's stored energy. Okay, so a cell, again, can just briefly, very briefly change the voltage, change the electrical potential on either side of its membrane. And here's a very simple GIF of how this action potential works. Hey, if this is the axon of a nerve, okay, take a look. It's positive on one side, negative on the other, just like a battery. And for a brief moment, it switches. That's all an action potential is. The passage of this signal, just telling parts of the cell to switch very briefly. Yeah, that's it. That's what all an action potential is. And this little shift in electrical potential, traveling down at very high speeds, at instantaneous speeds, that's the process by which the nervous system works. And how do we get these positive and negative charges on each side? Well, we have ions, which are charged molecules. In this case, we have sodium and potassium, which again are electrically charged atoms. All they do is just switch sides for a moment. Okay, so that's what an action potential is. And how is that going to be able to happen? Well, we got to start off somewhere, right? So what we're going to walk through is how that electrical potential just switches, how those sodium ions and potassium ions just kind of switch places very briefly. Okay, so neurons, they need to, well, in order to receive an action potential or to conduct an action potential, they need to receive a signal to do so. And if they don't receive the signal to do so, the neuron is what we call at resting potential. It's at resting potential. It means it's not firing any sort of signal. It's not firing an action potential. It's not switching the sodium and potassium just yet. Okay. Why we call it resting potential is because, well, there's a certain level of electrical potential energy that the neuron likes to stay at, and that is negative 70 millivolts. Negative 70 millivolts is the charge difference between the one side of the membrane and the inside of or the outside and the inside of the membrane. Okay, so negative 70 millivolts, like a battery is like 1.5 volts. You've seen 9-volt batteries before in the past. We're looking at the electrical potential difference of negative 70 millivolts. All right, here's how to remember what resting potential looks like. Okay, the outside of the cell, as we can see from this picture, I'm going to just move my face for a second. The outside of the cell, we can see these yellow dots. These are representing sodium ions. Sodium and potassium are the two big players here. Sodium ions are mostly found on the outside of the cell, and potassium ions, K+, are mostly found on the inside of the cell. Okay, so we got sodium ions and potassium ions, but hey, aren't they both positively charged? Well, there just happens to be more of these positively charged ions on one side than the other, thus resulting in a negative voltage. But a good way to remember what a axon looks like at resting potential is the phrase salty banana. It's really silly, but silly things help you remember sometimes. Okay, it's got sodium, which reminds you of salt on the outside, and potassium, which reminds you of potassium. Potassium, which reminds you of bananas on the inside. Okay, so our cell is at resting potential. It's not doing anything just yet. Okay, and there's a very special protein, a very famous protein. It's called the sodium-potassium pump that is going to be maintaining the resting potential of the neuron. And it requires ATP in order to operate. So what kind of transport is this? Well, we're dealing with a type of active transport. ATP comes into the picture, and what this sodium-potassium pump does, it forces 3 sodium ions out of the cell for every ATP molecule that it uses up, and it moves 2 potassium ions into the cell. Okay, so we're maintaining the salty banana by putting that sodium on the outside and the potassium on the inside. Okay, and so long as sodium's on the outside and potassium is on the inside, we'll probably stay right around negative 70 and stay at resting potential. Okay, so our sodium-potassium pump is going to come back into the picture later on. All right, so what we're looking at here, I'm going to be filling this out, and hopefully you have a copy of this. I'm going to be filling this out as we discuss the action potential here, and we're going to walk through each of the steps of this. So what we just got to was discussing resting potential, and there's our flat line here. We got membrane potential on the y-axis and time, which is like not even a fraction of a second, this whole thing, on the x-axis. All right, so our membrane potential goes way up and goes way down, then slowly comes back up over the course of every time a neuron has to send an electrical signal. Okay, but for right now, we haven't gotten there yet. We're at resting potential, or right here at nice and balmy negative 70 millivolts. Okay, so there's no stimulus yet. The neuron has not been told what to do. Okay, that's until, well, it does receive a stimulus. Okay, when the neuron does receive a stimulus, it starts to depolarize, and I'm going to be asking you to know what that word is. So depolarize, which means that it's going to become less polar on either side. Okay, so what's going to happen is that some sodium channels. Okay, so now we got to think about, why do we need sodium channels? Well, ions, which are positively charged molecules, ions can't cross the cell membrane by themselves. Okay, they can't just diffuse wherever they want to go. They need to go through a channel. Okay, so sodium channels start to open up. Maybe because of some stimulus, okay? So some sodium channels start to open up, and sodium starts to rush into the neuron. Okay, remember, we got a lot of sodium on the outside, not a lot on the inside. Okay, so where is sodium naturally going to flow? It's going to go right into the cell. Okay, so it's going to kind of start to rush into the cell once those sodium channels open. Okay, and like I said here, sodium cannot pass through without those channels. It's blocked by the cell membrane because it's a charged molecule. Okay, and when sodium comes into the cell, this is going to lower the voltage between the two sides, and it's going to depolarize. All right, so if you're decreasing the voltage, decreasing the difference, that's called depolarization. Okay, so how does that even begin? Well, Um, the sodium channels, many sodium channels are what we call voltage gated. Okay, so imagine this. Imagine a sodium channel like a door. Okay, and once you open the door, all the sodium can rush into whatever room the door goes to. All right, so you got sodium outside the door, and then and then you got your sodium channel, which is a door. Okay, you open that up, and the sodium channel comes in. Okay, that door is locked in many cases. So what's the key? The key here in this case is a change in voltage. All right, so it's voltage gated. A change in voltage kind of acts like a key for that door. So the voltage gated means that they will only open up if a certain voltage is reached. Okay, so it's like almost like a password, almost like, oh, did you reach threshold potential? I don't know. But that particular voltage that will serve as the key to open the door and allow the sodium channels, or excuse me, the sodium ions to go in, that is negative 55 millivolts. All right, so we need a very slight change, only a difference of 15 millivolts, in order to reach the threshold potential. Okay, so now if the stimulus has to be strong enough for this voltage to be reached in order for the action potential to even occur, or in order for that neuron to pick up that signal and send it. All right, it needs to reach negative 55. If it doesn't, the stimulus is not strong enough, then there is no action potential. Okay, so think about something that might be occurring around you, but you're not even going to be consciously aware of it. Okay, maybe that stimulus was not strong enough to trigger your cells to reach threshold potential. Excuse me, your neurons to reach threshold potential, and for you to process that. Excuse me, I'm getting a lot of notifications here. So if threshold potential is not reached, there's going to be no action potential, and your neuron's not going to do anything. But if it does reach negative 55, well, then we're going to start depolarizing. We're really going to start getting going here. All right, so negative 55, like I said, is our threshold potential. We're at resting until some kind of stimulus occurs. It starts to go up, but if it doesn't reach threshold potential, it's not going to pass through, and it's not going to cause an action potential. Okay, but once threshold potential is reached, sodium channels open, and they cause a lot of sodium to flood in, and that's where we're going to get our depolarization. In fact, there's so much sodium that rushes in from the outside to the inside of the cell that the voltage goes from negative 55 all the way to positive 30. And we have another term. We've talked about resting potential. We've talked about threshold potential. The third potential that we're going to be talking about is peak potential, which is positive 30 millivolts. Okay, so think back to that GIF that I showed you, positive, negative, just switch sides very briefly, right? We're about there. Okay, we're well into this. So we've started to depolarize. Okay, all that sodium is rushing into the cell up until the point that we reach positive 30 millivolts. Okay, and that's what we call peak potential. Yeah, so sodium is coming in. I'm going to add that over here. Na plus in. Ooh, I gotta change the font color. There we go. Na plus in. Okay, on the when depolarization is occurring. Okay, so once we reach peak potential, the neuron must now not depolarize but repolarize. And in order for that to happen, in order to try and bring the polarization back down, okay, because once we reach peak potential, that signal has been sent, and that's going to cause like the domino effect chain reaction down the axon, and your neuron is successfully sending a signal. It's successfully sending that electrical impulse. So at this point, the neuron must now repolarize, and potassium channels open at peak potential, causing repolarization. All right, so the reverse is happening now. Potassium ions now start flooding out of the neuron, causing the voltage to go back down. Okay, so our sodium channels opened up, the sodium channels rushed in. Now the sodium channels are closed. The door for sodium is now closed, and now the door for potassium is open. Okay, and now since we got a lot of sodium and a lot of potassium on the inside, once we open this up, potassium floods out of that axon, of that cell. All right, so that's where we're getting repolarization. Our voltage is going down from peak potential, and in fact, we go even lower we get even more, or I should say less voltage than at resting potential. Okay, so once we dip below resting potential, this is called hyperpolarization. Okay, and there's a slight delay in the opening, or I should say the closing of potassium channels. A slight delay in those potassium ion channels, the door for potassium to pass through to the other side of the cell, um, stays open just for a little too long, and it causes the voltage to go below resting potential. All right, so the potassium channels are still open, but once we reach about negative 80 millivolts, which is again negative 70 is resting, this is less than resting potential. Okay, then those potassium channels are going to start to finally close. Okay, but so we're back here, we're down here, we're at hyperpolarization when we're below resting potential. Okay, so now that we're below resting potential, what has to happen in order for us to go back to resting potential? Well, our friend sodium potassium pump comes back into the picture. Okay, we can't fire another action potential. The neuron literally cannot send another signal until it's back at resting potential. Okay, and so our sodium potassium pump is going to be using up a lot of ATP to try and bring us back to normal. That's why neurons and nerve cells are such a huge energy hogs. They use so much ATP because they have to maintain this resting potential at all times. So during repolarization and hyperpolarization, an action potential cannot occur again. And that's what we call the refractory period. So right down here, in this range, right before we get back to resting potential, that is called the refractory period. You can't fire another action potential until resting potential has once again been reached. All right, so sodium potassium pump is going to do that, and the neuron must go from negative 80 back to negative 70 in order to fire another action potential. Okay? So, oh, there it is. I highlighted it on the next one. So there's our refractory period. We can't fire another action potential. We can't depolarize again for this whole highlighted part here. Okay? Now, once the neuron returns to resting potential, another action potential can occur. The neuron can keep firing. Okay? Just keep in mind that this is not even a fraction of a second. This is like so fast. It's ridiculous. I don't even know the particular amount of time that it takes, but it's really fast. Okay? So take a look at this axon over here. What we're seeing here, how an action potential passes from one, does it go down the axon? Well, it kind of jumps from one node of Ranvier to the next. Okay? It kind of skips the part where it's myelinated. Okay? The action potential skips the part where it's myelinated. So an axon with more myelin at it will be faster at sending action potentials than one that's unmyelinated. Okay, so going back to that GIF one more time. Let me just bring it up quick so you know what I'm talking about. I'm going the wrong way. Here it is. Okay, this is an unmyelinated axon. See how slow it's going? Okay, it's got a reverse at every single one of these changes. Okay, imagine if it were able to go skip, 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 skip. Okay, that's what a myelinated axon can do. Okay, so that myelin sheath, what we looked at in our last video when we were taking a look at the, when we were taking a look at the, at the anatomy of the neuron, the myelin is really going to be helpful for insulating and helping transmit those signals faster. Okay, so this notice of Ranvier, that's where this switch is really going to be happening. Okay, and the other thing that's going to make a neuron better or action potential faster going from one point to the next is a larger axon diameter. It's going to be, so the larger the axon diameter, the faster the message is going to go through as well. Okay, so last term I want to introduce to you here, this is what we call the domino effect. Look, I brought it up again. I really should look at this before I make the video. So once one node of Ranvier undergoes the change in potential, it triggers another node to depolarize, and that's what's called the domino effect. Okay, so it's like dominoes. You topple one over, and it topples the rest of them over, right? So it goes from one, which triggers the next, which triggers the next, which triggers the next, so on and so forth. All right, rather than this unmyelinated axon over here. Okay, I believe that is it. Yeah, we're getting into the central nervous system in our next video, so please let me know if you have any questions, and we will see you next time.